Have you ever just looked around at the sheer variety of life on our planet and wondered, how on earth do we even begin to make sense of it all? From the massive trees in our forests to the microscopic organisms swimming around in a single drop of pond water, the incredible diversity of life on Earth presents a frankly massive sorting problem. Imagine trying to organize a library with millions of books, but half of those books are completely invisible to the naked eye. Well, that is exactly the monumental challenge scientists faced when trying to categorize the living world.
Okay, let's dive into this. Welcome to today's Explainer, where we're going to unpack the fascinating world of biological classification. We're going to see exactly how we went from basically just sorting things by what they look like to mapping out the incredibly complex, microscopic branches of the tree of life using structured categories. Let's unpack this massive biological puzzle together and see how scientists managed to bring order to all this chaos.
So here is our roadmap for today. We'll start with the history of classification, then look at the five-kingdom classification system. After that, we'll zoom in on Kingdom Monera, the world of bacteria, and wrap up with Kingdom Protista, looking at single-celled eukaryotes.
All right, Section 1, History of Classification.
So, long before we had high-powered microscopes or DNA sequencing, people just had to use pure logic to categorize life. Take Aristotle, widely known as the father of biology. He essentially looked out his window and logically divided the visible world strictly into two camps: plants and animals. Pretty straightforward, right? For plants, he sorted them by size: small herbs, medium shrubs, and large trees. For animals, he looked at where they lived, so on land, in the air, or in the water. But he took it one step further with a rather unique physical trait. He categorized animals based on whether they had red blood, which he called eneima, or lacked red blood, which he called aneima, like octopuses. It was a brilliant logical start based purely on what he could actually see.
But as science progressed, that simple logic kinda hit a wall. In 1758, Linnaeus formalized a two-kingdom system, Plantae and Animalia. But then a real game-changer came along: the microscope. Suddenly, scientists were seeing single-celled organisms everywhere that simply didn't fit into the plant or animal boxes. So in 1866, Haeckel added a third kingdom, Protista. By 1956, Copeland realized some of these single-celled organisms didn't even have a true nucleus, so he added a fourth, Monera. Finally, in 1969, R.H. Whitaker realized fungi were entirely distinct from plants, giving us the famous five-kingdom system. As you can see, the invention of new technology literally forced scientists to keep adding branches to the tree.
Moving right along to Section 2, the five-kingdom classification system.
This brings us to the globally recognized modern framework. Now, the absolute crucial point here is the shift from Whitaker to Woese. Whitaker's 1969 system categorized everything into five kingdoms based on clear traits like cell structure, body organization, and how they actually get their nutrition. But fast forward to 1990, and Carl Woese introduces an incredible update. He proposed a three-domain system, which effectively created six kingdoms. Why? Well, because he realized that the bacteria in Kingdom Monera were actually made up of two entirely distinct groups with completely different evolutionary relationships. So he just split them up into archaea and bacteria, keeping everything else safely inside the eukarya domain.
All right, Section 3, Kingdom Monera, the world of bacteria.
Let's follow Carl Woese's lead and zoom all the way down into that first massive microscopic grouping, the bacteria. Now, before we look at their diversity, we have to understand their basic building block. Kingdom Monera is made entirely of prokaryotic cells. Think of a prokaryote as a kind of primitive or poor cell. It lacks a true nucleus. Instead, its DNA is just floating around naked in the cytoplasm, in a region called a nucleoid. It also completely lacks membrane-bound organelles, so no mitochondria, no Golgi bodies, none of that. Interestingly, though, it does have 70S ribosomes to build proteins. Every single organism in Kingdom Monera operates with this incredibly simple but highly effective cellular machinery.
Now, while they are totally invisible to the naked eye, if you look under a microscope, bacteria actually have very distinct, recognizable shapes. They generally fall into four categories. First up, coccus bacteria. These are perfectly spherical. Think of Streptococcus. Second, bacillus bacteria. These are rod-shaped, like lactobacillus, which, fun fact, helps make your yogurt. Third, spirillum are spiral-shaped, like Helicobacter pylori. And fourth, Vibrio bacteria look exactly like tiny little commas, such as Vibrio cholerae. So even in the microscopic world, structure and shape are highly distinct.
Remember how Carl Woese split the bacteria into two groups earlier? And this brilliantly illustrates exactly why he did it. The extremes of this kingdom are wild. On one side, we have archaeobacteria. These are the ancient, incredibly tough survivors. Their unique cell wall composition lets them thrive in absolutely extreme conditions. We're talking thermoacidophiles living in boiling hot springs, halophiles surviving in totally extreme salt flats, and methanogens living in the oxygen-deprived guts of cows, literally producing methane gas. On the other side, we have eubacteria. These are the true bacteria with standard peptidoglycan cell walls. This group includes the everyday bacteria we encounter, from the ones that cause illness to the ones that photosynthesize.
Speaking of photosynthesizing eubacteria, let's look at one of the most important groups out there, cyanobacteria, also known as blue-green algae. These aren't just microscopic floaters, they are incredible problem solvers for the whole planet. Our atmosphere is 76% nitrogen, but plants can't use it in its gas form. Cyanobacteria, like nostoc, possess these specialized, thick-walled cells called heterocysts. Inside these oxygen-free heterocysts, they actually take that useless atmospheric nitrogen and convert it into nitrates and ammonia, forms that plants can easily absorb. They are literally nature's microscopic fertilizer factories.
But before we leave the bacteria behind entirely, we have to talk about nature's ultimate microscopic oddball, mycoplasma. These are officially the smallest living cells on planet Earth. But what makes them truly bizarre is that, unlike almost all other bacteria, they completely lack a cell wall. Because they don't have that rigid structure, they are highly adaptable shapeshifters. They can even survive entirely without oxygen. But unfortunately for us and for plants, this incredible adaptability also makes them highly effective pathogens that cause a variety of tricky diseases.
Section 4. Moving up the evolutionary ladder to Kingdom Protista, single-celled eukaryotes.
Okay, so we are leaving the primitive prokaryotes behind and entering a world with a major evolutionary upgrade, a complex, true nucleus. Within Protista, we find some spectacular marine organisms. First, the cruciferates, particularly the diatoms. They are the chief food producers of the ocean, and their cell walls are embedded with silica, making them virtually indestructible. They fit together exactly like a tiny soapbox. When they die, these silica shells pile up on the ocean floor, creating a gritty powder called diatomaceous earth, which we actually harvest for polishing and filtration. Now compare that to dinoflagellates. They have stiff cellulose plates and two flagella for spinning rapidly through the water. When certain types, like gonialax, multiply out of control, they cause what we call red tides, releasing toxins so powerful they can literally wipe out other aquatic life.
Yeah, what's really interesting about this slide is that some protists just refuse to play by standard biological rules. Take the euglenoids, for example. Instead of a rigid cell wall, they have a highly flexible protein layer called a pellicle, but their real magic trick is their diet. They employ what's called mixotrophic nutrition. In the presence of sunlight, a euglena is an autotroph. It uses its chlorophyll to peacefully photosynthesize its own food, exactly like a plant. But the very moment the sun goes down and they are in the dark, they completely shapeshift their behavior. They become heterotrophs, actively hunting and preying on smaller organisms to survive. Talk about getting the best of both worlds. It is a stunning, dual-natured survival strategy.
And just to prove how massive the variety of survival strategies is within Protista, we have the slime molds. Unlike the photosynthesizing diatoms or the shape-shifting euglenoids, slime molds are saprophytic. That means they are nature's microscopic recycling crew. They actively move across decaying twigs and dead leaves, engulfing and breaking down organic material. They are single-celled, sure, yet they gather together in the thousands to form visible, creeping masses that help keep our ecosystems clean.
So we've sorted through bacteria, we've explored the incredibly complex protists, and we've seen how scientists built these 5 intricate kingdoms to finally organize life. But looking at all of this structure leaves us with one glaring mystery. The 5-kingdom system clearly covers plants, animals, fungi, protists, and bacteria. Yet there is an incredibly famous, highly impactful entity missing entirely from this system: viruses. So why did viruses get absolutely no place in any kingdom? Are they alive? Are they dead? That is the ultimate biological exception, and exactly what we're going to explore in our next explainer. Thanks so much for joining me today, and keep questioning the world around you.